Hello, dear students. Today our lecture will be about human development, and we will discuss the following sections: fertilization, implantation, embryogenesis, development of placenta, maternal fetal blood circulation. Obstetrics, from the Latin obstrate, to stand by, is the field of clinical medicine that studies physiology and pathology of processes associated with conception, childbirth, the postpartum period, develops methods of obstetrics, prevention treatment of complication of pregnancy, childbirth, and assistance with them for a woman, fetus, and newborn. Perinatology or maternal fetal medicine is the branch of obstetrics that focuses on the medical and surgical management of high-risk pregnancies. Obstetricians who practice maternal fetal medicine are also known as perinatologists. This is a subspecialty to obstetrics and gynecology mainly used for patients with high-risk pregnancies. Obstetrics includes the following sections: physiology and pathology of pregnancy, childbirth and the postpartum period, operational obstetrics, physiology and pathology of fetus and newborn, which is now called perinatology. The human's ovary consists of 2 million oocytes at birth, and approximately 400,000 follicles are present at puberty onset. The remaining follicles are depleted at a rate of approximately 1,000 follicles per month until age 35, when this rate accelerates. Only 400 follicles are normally released during pregnancy reproduction life. Therefore, more than 99% of follicles undergo atresia through a process of cell death termed apoptosis. As you can see in the picture, in order to reach the ovulatory stage, an ovarian follicle will pass through the following stages. The first stage is primordial or resting stage. Second stage, primary or pre-antral stage. Third stage, antral stage. And the last stage, the pre-ovulatory follicle stage. We call it Graafian follicle. The menstrual cycle is a physiological process that fertile women undergo for the purposes of reproduction and fertilization. During menstrual cycle, some events happen in ovarian and uterus, and these events go parallel to each other. In ovarian cycle consists of follicular phase, ovulational phase, and luteal phase. Menstrual cycle consists of menstrual phase, proliferative phase, secretory phase, and premenstrual phase. The follicle phase starts on the first day of menstruation and ends with ovulation. Promoted by hypothalamus, the pituitary gland releases follicle-stimulating hormone. The hormone stimulates the ovary to produce 5 to 20 follicles with band on the surface. Each follicle houses an immature egg. Usually only one follicle will mature into egg, while others die. This can occur around day 10 of 28-day cycle. The growth of the follicle stimulates the lining of the uterus to thicken in preparation for possible pregnancy. The next stage is ovulation phase. It's the release of mature egg from the surface of the ovary. This usually occurs mid-cycle around 2 weeks or so before the menstruation starts. As you can see in the picture, during the follicular phase, the developing follicle causes a rise in the level of estrogen. The hypothalamus in the brain recognizes these rising levels and releases chemical called gonadotropin-releasing hormone. This hormone promotes the pituitary gland to produce raised level of lutein-rising hormone and follicular stimulation hormone. Within 2 days, ovulation is triggered by high levels of lutein-rising hormone. The egg is funneled into the fallopian tube towards the uterus by small waves, hair-like projections. The lifespan of typical egg is only 24 hours. Unless it meets a sperm during this time, it will die. Luteal phase. During ovulation, the egg bursts from its follicle, but ruptured follicles stay on the surface of ovary. For next 2 weeks or so, the follicle transforms into a structure known as corpus luteum. This structure starts releasing progesterone along with small amounts of estrogen. This combination of hormones maintains the signaling line of the uterus, waiting for fertilized egg to stick, to implant. If fertilized egg implants in the lining of uterus, it produces the hormones that are necessary to maintain the corpus luteum. This includes human chorionic gonadotropin and the hormone that is detected in a urine test for pregnancy. The corpus luteum keeps producing raised levels of progesterone that are needed to maintain the signaling line of the uterus. If pregnancy does not occur, the corpus luteum will wither and die, usually around 22 in a 28-day cycle. The drop in progesterone levels causes the lining of the uterus to fall away. This is known as menstruation, and then the cycle repeats again. And there are some events happen in the uterus parallel to ovarian events. First, it is proliferative phase. At the end of menstruation, the endometrium consists only of stratum basal. A new cohort of follicles develop in the ovaries. They secrete more and more estrogen. Estrogen stimulates mitosis in the stratum basalis and the prolific regrowth of blood vessels, regenerating the functionalis. By day 14 is 2 to 3 millimeters thick. Secretory phase, the endometrium thickens still more in response to progesterone from corpus luteum. The endometrium thickens as a result of secretion and fluid accumulation rather than mitosis. Progesterone stimulates endometrial glands to secrete glycogen. Glands grow wider, longer, and more coiled. By the end of this phase, the endometrium is 5 to 6 millimeters thick, a soft, wet, nutrient band available for embryonic development. Premenstrual phase. The corpus luteum atrophies and progesterone levels fall sharply, triggering spasmodic contraction of spiral arteries. It causes endometrial ischemia and brings about tissue necrosis and menstrual cramps. Pools of blood accumulate in stratum functionalis, and necrotic endometrium mixes with blood and serous fluid and forms the menstrual fluid. When enough menstrual fluid accumulates in the uterus, it begins to be discharged by vagina. In the ovary, follicle grow and oocyte mature, so it's ready for fertilization. At the same time, sex hormones are produced in the ovaries, which provide changes in the lining of the uterus, which is capable of receiving a fertilized egg. Stages of human development: fertilization, cleavage, gastrulation, organogenesis, maturation. childhood, puberty, adulthood. Development in humans lasts about 280 days or 40 weeks from fertilization until birth. Embriogenesis periods. Initial period, the first week of development. Embryonic period from the second week until the 8th week. Fetal period from 8th week up to the birth. First week of development. Fertilization, cleavage, and implantation. Fertilization, the process of fusion or union of spermatozoa with a mature ovum, is known as conception, fertilization, or impregnation. Fertilization is a natural life process which is carried out by the fusion of both male and female gametes, which result in the formation of the zygote. The male gamete is the sperm, and female gamete is the ovum. In humans, the process of fertilization takes place in the fallopian tube. During these processes, semen comprising thousands of sperms are inseminated into the female vagina during coitus. The sperms move towards the uterus and reach the opening of the fallopian tube. Only a few sperms will succeed in reaching of the fallopian tube. Stages of fertilizations are approach of sperm to ovum and agglutination, capacitation, agglutination reaction, penetration of sperm into the ovum and acrosome reaction, cortical reaction, activation of ovum, fusion of sperm and egg nucleus. Approach of sperm to ovum and agglutination. A single ejaculate of sperm contains around 200 to 400 sperms in approximately 3 to 3.5 milliliters of semen. However, only sperms are able to reach the fallopian tube. This is because of acidity of female genital tract. Some are engulfed by leukocytes of vaginal epithelium. Some are trapped in the cilia of the fallopian tube. Capacitation. The changes in the mammalian sperm that make it to fertilize the ovum are called capacitation. The main changes taking place are actimal motility, wiping movement of the tail with swinging movement of the head, alteration of acrosomal membrane, removal of glycoprotein layer for exposing the membrane receptors. Agglutination reaction. The ovum secretes a chemical substance called fertilizing. It is a glycoprotein that has a number of spermophilic sites on its surface for the anti-fertilizing presented on the head of sperm. The adhesion of spermatozoa to the surface of egg is brought about by linking of fertilizing and anti-fertilizing molecules. This reaction causes agglutination of sperm and sends out the number of sperm reaching the egg to reduce changes of polyspermy. The process of fertilization involves penetration of sperm into ovum, activation of ovum, fusion of sperm and nucleus. Penetration of sperm into ovum and acrosome reaction. The ovum, at the secondary oocyte stage, is extruded from the ovary along with corona radiata cells. These follicular cells are glued together by mucopolyhedride, the hyaluronic acid. The sperm provides enzymes that are known as sperm lysins. The hyaluronidase enzyme helps to dissolve hyaluronic acid polymers in the intracellular spaces. which hold granulose cells of corona radiata together. Corona penetrating enzyme dissolves the corona radiata, and ochrosine which dissolves the zona pellucida. The nucleus and the cytoplasm of sperm are drawn in the ovum. Due to this acrosomal reaction, the plasma membrane of sperm fuses with the plasma membrane of secondary oocyte, so that the contents of sperm enter the oocyte. This induces the depolarization of the oocyte plasma membrane and injures monospermy. Cortical reaction. Due to this acrosomal reaction, the plasma membrane of sperm fuses with the plasma membrane of secondary oocyte, so that contents of sperm enter the oocyte. After fusion of sperm with egg membrane, cortical granuloses release chemicals into the prevolatile space that harden the zona pellucida. This prevents polyspermy. Activation of ovum. The entry of sperm includes a chemical signal in the egg cell. This initiates the formation of fertilization membrane around the egg. The cortical granules present beneath the plasma membrane of oocyte fuse with the membrane and release their contents between the plasma membrane and zona pellucida. This hardens the zona pellucida and further prevents the entry of other sperm into egg cell. The secondary oocyte completes the second meiotic division, producing ovum and second polar body. The second polar body degenerates immediately. Fusion of sperm and egg nucleus. The point of contact of sperm entering the oocyte is called as a cone reception or fertilization cone. The sperm nucleus now moves towards the egg nucleus along a definite path called copulation path. This leads to the fusion of the male and female pronucleus called fertilization. The fertilized egg is now called zygote. Significance of fertilization is that it stimulates the secondary oocyte to complete its maturation to form haploid ovum. It restarts the diploid number of chromosomes in zygote. Significance of fertilization is that it activates the egg to develop into a new individual by repeated meiotic division. It becomes more metabolically and more active. Significance of fertilization is that it combines characters of two parents. This causes variation and help in evaluation. A zygote is the simplest form of life. So once zygote is formed, it proceeds to split and divide into cells that organize and form more complex structure. Around 30 hours after the zygote is formed, the process of mitosis begins. Zygote contains 46 chromosomes, 23 the egg and 23 from the sperm. Cleavage of zygote. Around 30 hours after the zygote forms, the process of mitosis, cell division, begins, which increases the number of cells without any increases in zygote's total size. This is possible because with every division, the size of cell becomes smaller and the number continues to double. This process is called cleavage, and the cells of the dividing zygote are called blastomers. At this stage, the zygote is covered with an outer covering called zona pellucida. So it's divided rapidly by mitosis to produce 2 cells, then 4 cells, then 8, 12, and etc. And continues to divide. Once the zygote reaches the 8-celled stage, the blastomers arrange themselves tightly against each other. This is called compaction. When there are 12 to 32 plastomers present, the structure resembles a mulberry and it's now called a marula. At this point, developing individuals is referred to as marula. The cells of the marula give rise to the cells of the embryo, as well as cells that form the structure supporting developing embryo. 4 days after fertilization, the marula moves into the uterus and the fluid-filled cavity is formed. Marula form a hollow ball of cells called blastocysts. As the fluid in the fluid-filled blastocyst increases, two types of cells are seen, the inner cells and trophoblast cells. The inner cells, called inner cell mass, or also can be called embryoblasts. The inner cells of the blastocysts will eventually form the embryo. These cells are not yet specialized. They have a phenomenal ability to differentiate and divide to give rise to many different types of tissue. Trophoblasts, the outer layer of the blastocyst, forms. This will later develop into the layer of membranes that surround the embryo, placenta and amnion. Let's take overview. In the ovary, as you can see in this picture, follicle passes through the stages primordial, primary, tertiary, and graphial follicle stage. In the oviduct, fertilization starts. The egg is viable for 12 to 24 hours after ovulation. Sperms are viable for 12 to 48 hours after ejaculation. Sperm must make their way to uterine tube for fertilization to be possible. So fertilization occurs in the fallopian tube, as you can see in the picture. Sperm and egg join. They form zygote. Zygote begins rapid mitotical cell division. Next stage start cleavage. After embryo floats free to the uterus. Uterine secrets are used for nourishment. By the 10th day of the blastocyst is completely embedded in the endometrium. Blastocyst enters the uterus and here it will implant to the uterus wall. The endometrium now provides nourishment for the developing blastocyst. Connection with the mother will begin to form, placenta and umbilical cord. For 6 or 7 days after fertilization, the embryo implants the uterine wall. This process can be divided into 3 phases. Apoptosis, initial contact of blastocyst to the uterine wall. Adhesion, increased physical contact between the blastocyst and uterine epithelium. Invasion, penetration and invasion of syncytiotrophoblasts and cytotrophoblasts into endometrium, inner third of the myometrium, and uterine vascularity. Successful implantation requires a receptive endometrium, appropriately primed with estrogen and progesterone by corpus luteum. By the time of its interaction with endometrium, the blastocyst is composed of 100 to 250 cells. The blastocyst loosely adheres to the endometrium epithelium by apoptosis. This most commonly occur on the upper posterior uterine wall. The development of fully formed organism form a fertilized egg, embryogenesis, 3rd week of development. Gastrulation, its formation of 3 germinal layers. Within 20 days, embryonic membrane become clearly distinguished from embryo. The amnion is a thin membrane filled with amniotic fluids that eventually surround embryo and act as shock absorbent. Later, allantation membrane develop, which develop towards the chorion and get fused to form allantochorion, which later form placenta. Yolk sac has no significant function in humans. Embryonic disc present between yolk sac and amnion give rise to embryo. Embryonic disc differentiate into three germ layer: endoderm, mesoderm, and ectoderm, known as gastrulation. Embryo show distinct from at about 4 or 5 weeks. Only after 6 weeks, embryo can be distinguished as human embryo and now embryo term as fetus. Each of these layers give rise to specific structure in the developing embryo. Embryonic ectoderm give rise to the epidermis, central peripheral nervous system, eyes, internal ears, neural crest cell, and many connective tissue of head. Embryonic endoderm is source of epithelial lining of the respiratory and digestive tracts, including the glands opening in the gastrointestinal tract and glandular cells of associated organs such as liver and pancreas. Embryonic mesoderm give rise to all skeletal muscles, blood cells, the lining of blood vessels, all visceral smooth muscular coats, serosal linings of all body cavities, ducts, and organs of the reproductive and excretory system, and most of the cardiovascular system. In the body, excluding the head and limbs, it is the source of all connective tissue, including bones, tendons, ligaments, dermis, and stroma of internal organs. As blastocyst reaches to the uterus, trophoblast cells invade the endometrium wall and utilize the nutrients for its growth and multiplication. This invasion establishes the embryo within 6-9 days in the uterus, called implantation. With successful implantation, trophoblasts form a chorion membrane, which later become a part of placenta. Chorion membrane develops small wheel-like projections on its outer layer called chorion villi that begins to grow in the endometrium and help in exchange of nutrition between embryo and uterus. Embryonic cells grow to become embryo. It also forms other embryonic membrane covering the embryo. Neurulation. Neurulation. Embryo results in the formation of neural type, which gives rise to both the spinal cord and the brain. Neural crest cells are also created during neurulation. Neural crest cells migrate away from the neural tube and give rise to a variety of cell types, including pigment cells and neurons. Neurulation begins with the formation of neural plate and sinking of the ectoderm caused when cuboid epithelial cells become columnar. Changes in the cell shape and cell adhesion cause the edges of the plate fold and rise, meeting in the midline to form a tube. The cells at the tips of the neural folds come to lie between the neural tube and the overlying epidermis. The cells become the neural crest cells. Both epidermis and neural plate are capable of giving rise to neural crest cell. In this slide, you can see the human development milestones by the age and week gestation and external characteristics of embryo. Chorion wall surrounds the chorion cavity and forms villi. primary chorionic villi, cytotrophoblast, syncytotrophoblast, secondary chorionic villi, extra-amniotic mesoderm, cytotrophoblast, syncytotrophoblast. By the 9th day, lacuna develop in the syncytotrophoblast. By the 12th day, the dispersed lacuna form a lacunar networks. Meanwhile, endometrial capillaries form the sinusoids. Blood will flow between the sinusoids and the lacunar networks forming the uteroplacental circulation. Development of chorionic sac. Primary chorionic villi begin to appear by the end of the 2nd week, induced by the extraembryonic somatic mesoderm. Extraembryonic somatic mesoderm plus cytotrophoblast and syncytotrophoblast make chorion walls of chorionic sac. The extraembryonic columella become chorion cavity. Column become chorion cavity. Development of the chorionic villi. Primary chorionic villi turns to secondary chorionic villi, tertiary chorionic villi. Cytotrophoblastic cells proliferate and form cytotrophoblastic shell that surrounds the chorion and attach it to the endometrium. Exchange occurs between the embryonic blood in the branchiole of the tertiary chorionic villi and the maternal blood in the intervillular spaces. Development of placenta. The villus chorion will form the fetal part of placenta. The deciduo basalis will form the maternal part of placenta. The placenta will grow rapidly. By the end of the 4th week, the deciduo basalis is almost entirely replaced by the fetal part of placenta. As blastocyst implants itself the uterine wall and the placenta begin to form, the corpus luteum is nearing the end of its programmed 12-day life cycle. Unless developing embryo sends a signal, hormone levels drop and the embryo will be flushed from the body. Placenta secretes hormones that inhibit menstruation. Placenta, tree of life. The derivation of the word placenta comes from the Latin for cake, from Greek for flat, slab-like. Structurally, placenta is a hemorrhagical villus organ. Functionally, the placenta is a highly complex machine. It acts like a lung in the exchange of oxygen and carbon. It works as a digestive system, absorbing all necessary nutrients for fetal development and growth. It functions as a kidney to remove waste, and it behaves as immune barrier to protect the growing fetus from antigen attack from the maternal system. The placenta is also an important endocrine organ, producing many hormones and growth factors that regulate the course of pregnancy, support and promote fetal growth, initiate parturition. However, all this depends on normal vascular development within placenta itself. Normal placenta vascular development ensures a healthy pregnancy outcome, whereas insufficient of abnormal placenta vascular development will compromise pregnancy outcomes, both of the mother and the fetus, with complications that include preeclampsia, intrauterine fetal growth restriction. Thus, the main functions of placentas are protection, nutrition, respiration, excretion, and hormone production. The placenta is attached to the uterine wall and establishes connection between the mother and fetus through the umbilical cord. It has two components. Fetal part developed from the Hornig sac. Maternal part derived from the endometrium functional layer of the pseudobasalis. The placenta is a unique vascular organ that receives blood supplies from both the maternal and fetal systems, and thus has two separate circulatory systems for blood, the maternal placental blood circulation and fetal placental blood circulation. Deciduum is a functional part of endometrium that will be expelled after labor. This is the endometrium of gravid uterus. It has four parts. Deciduobasalis. It forms the maternal part of placenta. Deciduocapsularis. It covers the conceptus. Deciduoparitalis, the rest of the endometrium. Deciduoreflection, junction between the capsularis and paritalis. At around the 4th month, the fetus is so large that deciduocapsularis comes into contact with deciduoparitalis. The merging of these two deciduo causes the uterine cavity to obliterate. The placenta. At the end of 20th week, the placenta is enlarged, the amnion fused with the Hornig sac forming amniohornig membrane. The deciduocapsularis degenerate and the amniohornig membrane adhere to the deciduoparitalis. In the full-term placenta, cytotrophoblastic shell will anchor to the fetal placenta to the deciduobasalis. Placental septa will develop from deciduobasalis towards the Hornig plate, dividing the fetal placenta into cotyledon. Each cotyledon contains 2 or 3 stem wheels, which are surrounded by the intervillous spaces that develop from the lacunar networks. Each stem Hornig wheel contains many branch wheels. Hornig wheelie contain fetal blood vessels that is branched from branch wheel into the Hornig plate, which are branched from the umbilical branch wheels. Exchange happens through the placental membrane, which consists from cytotrophoblast, cytotrophoblast connective tissue, capillaries, and endothelium. Cytotrophoblastic cells begin to disappear, and then capillaries come in direct contact with cytotrophoblast. Exchange of oxygen and nutrients take place as the maternal blood flows around terminal villi into the intervillous space. The inflowing maternal arterial blood pushes the oxygenated blood into the endometrial and then uterine veins back to the maternal circulation. The fetal placental circulation allows the umbilical arteries to carry the oxygenated and nutrient-depleted fetal blood from the uterus to the villus from core fetal vessels. After the exchange of oxygen and nutrients, the umbilical veins carry fresh oxygenated and nutrient-rich blood circulating back to the fetal systemic circulation. Maternal blood flow to the placenta is approximately 600–700 milliliters per minute. It estimated the surface area of cytotrophoblasts is approximately 12 meters square. The length of fetal capillaries of fully developed placenta is approximately 320 kilometers at term. The functional unit of maternal-fetal exchange of oxygen and nutrients occur in the terminal villi. No intermingling of maternal and fetal blood occurs in placenta. In the picture in the right panel shows the relationship of the uterus, placenta, and fetus during pregnancy. The left panel shows the directions of blood flow from mother to placenta, and fetal blood flow from the placenta to fetus placenta, provides the fetus with oxygen and nutrients, and takes away wastes such as carbon dioxide via the umbilical cord. The extra-embryonic membranes. There are 3 layers of tissue that surround the growing embryo. These are extra-embryonic membranes that provide protection and nourishment to the developing fetus. These are amnion, develops above the embryo. It protects the embryo from injury. The amnion and the amnion cavity enlarge and surround the embryo. Allantosis, the cavity that develops as an outgrowth from the hindquarter. Horion, it completely surrounds the developing embryo. It is composed of trophoblast outside and mesoderm on the inner side. The lining of allantosis fuses with the somatic mesoderm of the horion to form the allantohorion. These layers then develop to form placenta. Yolk sac develops below the embryo. Its wall is made of endoderm and mesoderm. It serves as site of early blood cell formation until 6 weeks when the liver takes over this role. Amnion consists of amnion sac that is filled with amnion fluid. The amniotic sac attached to the embryonic disc, and with the folding on embryo, it surrounds the embryo, attaching to it ventrally and covering the umbilical cord. The amnion enlarges, obliterating the horion cavity, and come in contact with horionic sac. Amnion is thin protective membrane that surrounds the embryo. Start developing in the early 2nd week after fertilization as an enclosed cavity in the embryoblast. This cavity is roofed in by a single layer of flattened cells, the amnioblast, and its floor consists of the epioblast of the embryonic disc. Outside the amniotic ectoderm is a thin layer of extra-embryonic mesoderm, extra-amniotic mesoderma amniotic, and ectoderma amniotic cavity epioblast. Amnion cone is attached to the margins of the embryonic disc. As the embryonic disc grows and folds along its margins, the amnion and the amnion cavity enlarge and entirely surround the embryo. From the ventral surface of the embryo, it is reflected on the connecting stalk and thus forms the outer covering on the future umbilical cord. The amniotic fluid increases in quantity and causes the amnion expand. The amnion ultimately adheres to the inner surface of the horion, so that the horion cavity is obliterated. The fused amnion and horion form the amniohorionic membrane. Further enlargement of amniotic cavity results in obliteration of uterine cavity and fusion of amniohorionic membrane with the decidual parietalis. Amniohorionic membrane are usually ruptured just before the birth. Amnion fluid is a clear, slightly yellowish liquid. 99% of fluid in amniotic cavity is water. Suspended in this fluid are indissolved substances: desquamate fetal epithelial cells, proteins, carbohydrates, fats, enzymes, hormones, and pigments. As pregnancy advances, the composition of amniotic fluid changes as fetal waste products, meconium and urine, are added. Amniotic fluid circulation. The water content of amniotic fluid changes every 3 hours. Large volume moves in both directions between the fetal and maternal circulation, mainly through the placental membrane. It's swallowed by fetus, is absorbed by respiratory and digestive tract, and enters the fetal circulation. It then passes to the maternal circulation through placental membrane. During final stages of pregnancy, fetus swallow about 400 milliliters of amnion fluid per day. Excess water in the fetal blood is extracted by fetal kidneys and returned to amniotic sac through the fetal urinary tract. By the beginning of the second trimester, the amniotic sac contains 20 milliliters of amnion fluid. The volume of amnion fluid increased gradually, reaching about 1,000 milliliters by 37 weeks. High volume of amniotic fluid called polyhydramnion. It results when the fetus does not swallow the usual amount of amnion fluid. Low volume of amniotic fluid less than 400 milliliters is called oligohydramnios. The function of amniotic fluid is the fetus floats in the amniotic fluid. It allows fetus to move freely, aiding development of muscles and bones, prevent adherence of amnion to the embryo, act as cushion to protect embryo from injuries, act as barrier to infections, permits normal lung development, permits symmetrical external growth of the embryo, regulates fetal water electrolyte balance, assists in regulation of fetal body temperature. Umbilical cord connects the fetus to the placenta. The umbilical cord is formed from the fetal membranes, provides a circulatory pathway that connects the embryo to hornic villi of placenta. Its function to transport oxygen and nutrients to the fetus from placenta and to return waste products from the fetus to placenta. Measurement is about 53 centimeters. Develops from connecting stalk. The connecting stalk initially attached to the caudal end of the embryonic disc. After folding, becomes attached to the ventral surface of the curved embryonic disc at the umbilical region. The umbilical region, wider initially, becomes narrower as the folding progresses. The underlying structures are compressed together and form a cord-like structure, the umbilical cord. The umbilical cord is a lifeline that attaches the placenta to the uterus. During prenatal development, the umbilical cord comes from the same zygote as fetus. The cord is not directly connected to the mother's circulatory system. Instead, it joins the placenta, which transfers materials to and from mother's blood without allowing direct mixing. The umbilical cord contains one vein and two arteries, the umbilical vein, the umbilical arteries, buried with Wharton jelly. The umbilical veins carries oxygenated, nutrient-rich blood from placenta to the fetus, and the umbilical arteries carry deoxygenated, nutrient-dependent blood from the fetus to placenta. Any impairment in blood flow within the cord can be a catastrophic event for the fetus. The number of veins and arteries in the cord is always assessed and recorded at birth because about 1% to 5% of infants are born with a cord that contains only a single vein and artery. The rate of blood flow is 350 milliliters mean at the term. Blood can be withdrawn from the umbilical vein or transfused into the vein during the intrauterine life for fetal assessment or treatment. The cord is unlikely to twist or not to interfere with the vital oxygen supplies. In about 20% of all births, a loose loop of cord is found around the fetal neck at birth. The walls of the arteries are lined with smooth muscle. It contains no nerve supply. The act of expanding the fetus after a complete term of pregnancy or gestation is parturition. The gestation period is complete in 280 days. Parturition involves forceful muscular contraction called labor. The head of fetus settles down into the pelvic cavity 2 weeks before birth. When gestation is completed, labor starts and the fetus is expelled out. The onset of labor commences with the rupture of amniotic sac and leakage of amniotic fluid. The labor ends with the birth of placenta and amniotic sac membranes.